my face. When I'm looking to the side, I'm actually looking at a larger screen rather than my small laptop screen. And I'd like to thank the organizers for this opportunity for the Chemistry International webinar to talk about our project, one of our projects, which is plasmonically-enhanced ultrasensitive epitope-specific serologic assays for antibodies to SARS-CoV-2 spike protein in patient plasma. These are my disclosures, and it's sort of a mandatory slide from our university. And both Srikanth and I have various grants from NIH. We're among founding members of Origin Bioscience, which is a faculty startup. We and Washington University may receive royalties based on licensing of plasmonic flora from our faculty startup. And new this year for our disclosures is disclosing that both of us are on editorial boards that we receive remuneration for these editorial activities. And up front, pictured is Dr. Singamaneni, who's a full professor at Washington University. And the work I'm going to talk about is the result of 4 very gifted graduate students who have gone on to get their PhDs and are in academia and in industry. Now, as you all know, SARS-CoV-2, which gives the COVID-19, is a pandemic that's worldwide. And at the time this slide was made, there were millions upon millions of cases worldwide, millions of deaths. There are 5 variants of concern that's monitored by the World Health Organization, and SARS-CoV-2 is the 7th identified coronavirus affecting humans. In recent history, SARS-CoV-1 and MERS are caused epidemics. Now, our technology is based on what originally started as a plasmonic floor patch, which is basically a plastic skin that was studded with gold nanorods, and the skin was very versatile in the sense that you could lay it as a cover slip over whatever was going to give some fluorescent signal, and this was amplified thousands of fold. But to make things a little bit more versatile, we went to a fluid-based plasmonic floor with, at the heart is a gold nanorod, and there's a 2 to 3 nanometer siloxane polymer that's put around this gold nanorod to prevent quenching of fluorescent signals if the fluorophore was on the surface of the nanorod. As the siloxane polymer thickness increased, then the signal would degrade or decline. So it's very instrumental to have a precise 2 to 3 nanometer siloxane polymer, which was then decorated with bovine serum albumin that was derivatized with biotin, and then a fluorophore commercially available 800 CW was put onto this so that you had this enhanced fluorescent structure. Now, this is the spectra of the plasmonic floor, but you could see that without the gold nanorods and the siloxane band around the gold nanorod, you, on a relative scale, you had very little fluorescence, but combined in plasmonic floor, you could achieve 6,000-fold increase in your fluorescence. And we could tune this across the spectra so that we could use any dye from 400 to 800 and even higher wavelength. in order to get a fluorescent signal. So this enables multiplexing by using different fluorescent dyes. Now, the advantages of measuring epitope-specific antibodies are several, because the vaccine responses or an actual infection by whatever virus gives a polyclonal response, which is a sum of the neutralizing and non-neutralizing monoclonal antibodies that are elicited by the infection or the vaccination. And there's individual differences in the immune response, and you'll see that in a little bit. And you really need to know if vaccines are truly preventative and if they are covering evolving variants, because that has become a problem globally. And epitopes outside what's termed the receptor binding domain of the spike protein of the virus can neutralize viral infection, as well as epitopes inside the receptor binding domain, and these contribute to the overall virus neutralization. Now, this is sort of a thumbnail sketch in terms of the virus. Antibodies that elicit by the virus can bind to the surface of the virus, and this is sort of a stylized depiction of the spike protein. And people are typically concerned about antibodies into what's called the receptor binding domain, which is this region up at the top of the stylized spike protein version. But linear epitopes of amino acids 553 to 570 and 809 to 826 bind to surface regions of the spike protein, but they also can neutralize the ability of the virus to infect a host or minimize the effect of the viral infection on the host. So what we did is, these are the amino acid sequences of these two different linear epitopes. These were chemically coupled to bovine serum albumin as a scaffold, and we spotted small amounts of the BSA with the different epitope attached to them at the bottom of 96 well plate. The idea was that these BSA as a scaffold with the different epitopes, amino acid sequences specific to the virus, would then bind antibodies in patient plasma. We could use a second antibody to, let's say we're looking for IgGs or IgM or IgAs, would then bind to the antibodies elicited within the patient, and then with plasmonic floor, then we could measure these. And this is an example of what's called convalescent plasma. These were actually patients who were infected by the original Wuhan version of the virus, and you could see with dilution of the plasmas that different patients responded differently to have antibodies, in this case IgGs, to the spike protein subunit 1. And this is for spike protein antibodies, and this is to the nucleocapsid protein of the virus. And these are the antibodies that most of the antigen assays are measuring in people. But the take-home message is that different people respond differently, and they can raise a high titer of antibody to the different proteins or less of a titer, and that most people will have a very exuberant response to the nucleocapsid protein, so that's why it's in these antigen assays. Now, this is if you did a fluorescent-based ELISA for antibodies to the spike protein, and this is the Plasmonic-enhanced. This fluorescent, these were actual side-by-side comparisons of the same ELISA plate where we used a conventional CW800 fluorescent indicator, and you can see that no real signal was detected, but adding plasmonic fluor to the same series of dilutions, you can see that there was a nice fluorescent response, which was linear with time, independent of the dilution, or was linear with the dilution with time of the different assays. So again, this is an expanded version of what was shown previously, that we're looking for antibodies that would bind to these specific linear epitopes within the spike protein, which other people have found to be able to neutralize the viral infection. And a lot of different labs had come up with different regions of the spike protein, some within the receptor binding domain of the spike protein, and others outside the binding domain of the spike protein, which were found to neutralize viral infection. And within this region, we had our specific amino acid sequence, and within this region, our specific amino acid sequence. There's an abundant literature that define these epitopes that are neutralizing. So this is, as we'd seen before, we spotted 2 microliters of BSA that was derivatized with epitope 1 or epitope 2 at the bottom of standard 96 well plates. And you can see then this is the dilution of convalescent plasma from different patients. And again, you see that patients can have a different response in terms of their ability to raise an antibody against epitope 1 or epitope 2, and it will differ from patient to patient. And here you see that patient 15 had a nice response. These are duplicates assays within each lane here. For epitope 2 antibodies, not much for epitope 1, but here patient 17 had a nice response for antibodies for epitope 1 and not epitope 2. And the control plasmas here were pre-pandemic plasma samples that we had in the freezer from other studies. And this is a log scale for the response. And again, it highlights the fact that different patients have different responses to different epitopes within the spike protein. And then this is a whole well assay, and again it highlights the fact that if we just used a fluorescent-based assay for epitope-specific antibodies, the response was very meager or blunted. But if we use plasmonic fluor enhanced assay, you can measure the dilution of epitope-specific antibodies. And this was all within a 20-minute overall assay where there were 5 minutes to each step in terms of binding of the plasma dilutions, washing, then binding of an anti-human IgG, washing, binding of fluorophore or plasmonic fluor. And then down here is depicted the same dilutions of patient plasma in the standard HRP colorimetric assay. And this was again a 20-minute assay, so the binding of the original patient plasma was for only 5 minutes. And for those of you who run these colorimetric assays, remember that the color background rises with time, so that if you incubate for longer than 5 minutes in each step, let's say do a standard 4-hour overall assay, you have an increasing rising background, but the fluorescent assays or the plasmonic fluor-based assays, the background is the same. Now, the versatility of this is that most people are measuring the IgG class of immunoglobulins, but you know that different immunoglobulins arise in a specified manner upon infection. And here we're showing that the versatility of the assay is that you can also measure IgG or IgA, and within the IgA class, we could measure IgA1 versus and show that, again, different patients can respond that are epitope specific, and they can have an IgA response, which in this particular patient is an IgA1 response, and small amounts of total IgA can be realized as small amounts of the IgA1 response. And again, this is log scale showing the IgG in epitope 1 and epitope 2 from patient to patient, and the IgA to epitope 1 and epitope 2 from patient to patient. And this was all done with 1 to 500 dilution of patient plasmas. Now, the advantages, again, of measuring the epitope-specific antibody is that a vaccine response is polyclonal. So it's the sum of virus-neutralizing and non-neutralizing monoclonal antibodies, and we're really not concerned about the non-neutralizing antibodies. So most of the commercial assays are looking at IgG and measuring the total response. And the only meaningful response, or the truly meaningful response, is to specific epitopes on the viral protein that will blunt or actually abrogate any infection. Now, you can measure different IgG and IgA subclasses because some patients have an adverse response or a very adverse response to infection, and patients also can have an adverse response to vaccination, and this may be due to different subclasses of IgG or IgA within that response, and this will tell you if there's an adverse immunoglobulin response to infection or vaccination, and this may allow information to the treating physician to possibly minimize these adverse responses. Now, different vaccines have different efficiencies, even to the same virus strains, and you're familiar with Pfizer and Moderna vaccines having better than 90% response, whereas Sinovac is about a 50-50 response in terms of being able to blunt adverse reactions to viral infection. Now, of emerging importance is that SARS-CoV-2 variants of concern may escape current vaccine coverage. So you're familiar with the fact that the Delta variant came into prominence in 2021 and the Delta Plus back then. And now in 2022, the concern is about the Omicron variants ranging from the BA1 to the BA5 variant. And this is just a quick snapshot from something in Nature Medicine that shows within South Africa, studies found that in July to September of 2020, the original variants were the major cause of daily 7-day moving average infections. And then as it mutated to the Alpha, the Delta, and now the Omicron variants, these variants of concern are supplanting the original variants in terms of infecting people. Now, the plasmonic floor-enhanced assay in some gives you epitope-specific platform, and this can be rapidly adapted to any infectious disease, and it can be adapted to a lateral flow assay because remember, most of our studies were basically looking at 5-minute incubation of patient plasma with the capture epitope-specific amino acid sequences. And lateral flow assays are 10, 15, 20-minute quick assay. And so plasmonic floor is instrumental in allowing uncovering of these epitope-specific antibodies. So I thank you for your attention on all this. And if you have any questions, I'll entertain those. Again, thank you.